during surgery. It's going to come to us. We're going to do a routine workup on it, and then.
Julia is over with.
Oh, she's the only other one. Okay. And then, well, there's a couple different steps to pathology. It's a pretty labor-intensive process. So the first thing that happens is the organ comes to us. First thing we have to do is what's called a gross description, gross dissection. So it's basically like when you dissect anything here in school. We're going to cut it open, we're going to measure it, we're going to weigh it, we're looking for certain tissue structures. Where there's a tumor, we want to identify where the tumor is. We want to cut pieces of it out so that we can identify how close to a margin it is. Anything that we cut up then and submit for pathology goes through another step called processing. What that does is using heat and vacuum, we're going to take all of the water out of the tissue. We want to completely dehydrate the tissue. Depending on the size of the tissue, it can take anywhere from 1 hour to 12 hours. So if it's a tiny little GI biopsy, we don't want to subject it to a really long dehydration process. It's something fatty like breast, we want it to take a little bit longer. It's going to take longer to get that water out. So what happens when we take all of that water out, we're left with a bunch of tiny little holes in the tissue. We're going to fill that with liquid wax. So at the end of that process,
let me go check to see.
the next step is called embedding. So what we're going to do is we're going to make a mold out of that tissue. So we're going to make a little square mold out of the same wax, so it's the same consistency inside and out. The next step after that hardens is to make a slide. So we have an instrument called a microtome, which is essentially like a deli slicer. It's kind of the same idea. There's a blade. The tissue is going to go into a holder, and we're going to turn a wheel, and it's going to cut slices. We use wax, which is slightly sticky. So in theory, when it works properly, each section is going to stick to the next section. You're going to get what's called a ribbon. That's going to lay out on some water. And then we're going to use a glass slide to pick that up and put it on the slide. At that point, the tissue is clear. But we want to see what's going on inside the cells. So it's just much good to be clear. So we're going to apply dyes to it.
Hey, can we stop you guys for a moment?
Yeah, sure.
I want to introduce the senator to you. And
So you doing?
Hi.
Good.
this is Allison from Harcum College.
Hi, nice to meet you.
Brian on it.
Hi, nice to meet you.
And what are you guys learning?
Staining. The steps of histology.
Yeah?
Yeah.
Okay. Very good. So yeah, they're going through a staining process and being able to take samples of anything and look at cells and proteins. So we have a full histology lab here. It's in the other room. Some of it we do here. For 4 years in a row now, we've been recognized as the only high school in the country to have a histology lab. So this is very unusual. It might not look like much now. It's very unusual to happen. Yeah.
Thank you. Nice meeting you all.
Thank you.
Nice crowd.
Thank you. I forgot I was wearing this. Pictures and everything.
Everyone like staring. Wow.
Did you tie dye your outfit?
Yeah. That was crazy.
You might have to start doing that.
You look like a scientist.
Thank you.
Okay, so where did I leave off? Okay, so the cells are clear, right? Is that where I left off? Okay. So everything gets what's called a hematoxylin and eosin stain. We do that to every single slide that we cut. So we apply a dye called hematoxylin, which is plant-based. It comes from a logwood tree down in Brazil. So all the deforestation down in Brazil is a little unnerving to the histology people. But what that does is it stains the nuclei of the cells. And then eosin is the other part, and it's going to stain, it actually is an orange dye, but it's going to stain 3 different colors. It's going to stain a magenta, it's going to stain orange, and it's going to stain pink. It's staining cytoplasm, blood, and muscle fibers. So a doctor is then going to get those slides. So all the work we've done up until this point doesn't involve a doctor. They come in at the last minute and make the final diagnosis. The technologists are the people who do all the work behind the scenes. So an interesting fact about lab work is that 60 to 70% of medical decisions are based on lab results. So while most of us are relegated to basements or some hidden area of the hospital, I'm fortunate enough to have windows where I work. I overlook the dumpsters, but we have windows nonetheless. We're pretty important in the grand scheme of determining how a patient's going to be treated. The field of pathology is moving more molecular as personalized medicine becomes more and more prevalent. We've moved into identifying genes in tumor cells to determine how patients are going to react to certain chemotherapies or different immunotherapies out there. So I'm sure you've all seen commercials for Keytruda and Opdivo and medications like that. All of those results are based on histology testing. So we're able to isolate the PD-L1 antibody or the PIK3CA antibody in tissue, which will then guide a patient's treatment. So it's a pretty interesting thing that we're doing in histology these days. So I have two different stains for you guys to do. One, we're going to be looking for fungus in tissue, and the other one is called a trichrome stain. So I've started it for you just because the first step takes an hour. So it's sitting in this solution here. This is called Bouin's solution. It's picric acid, acetic acid, formaldehyde, it's a bunch of chemicals mixed together. Pathology is actually the most hazardous department in the entire hospital. If you like chemistry, histology is a great place to go. I hated chemistry in college, but this is very surface overview of chemistry. But I did not.
I don't like putting math and like, whoa. So many forms. We did cool stuff, but the actual paper and pen.
Yeah. It's just kind of understanding how chemicals react together with the fungus. What we're doing, the first step is an oxidation step. And so everything is fixed in a formaldehyde solution. We want to oxidize those aldehydes. To free up an extra aldehyde group, because the chemical that it's going to go into is going to bind to that aldehyde group. So it's kind of chemistry like that you need to know. But again, if you just know the steps, it's kind of like cooking, kind of just follow the recipe. But you need to be able to troubleshoot and figure out why things didn't work the way they worked. All right, so fungus, go back to fungus. We're going to use a solution called periodic acid. Like I said, it's going to oxidize the aldehyde groups. It's going to open up a free aldehyde group on the fungal cell walls. It's going to go into a reagent called Schiff reagent. That's going to bind to that. It's going to turn pink, which is going to then make the fungus pink. We're going to counterstain it with green, so anything that's not fungus is going to stain green, and that's what's going to look at the microscope. Going back to the Bouin solution, so what it's doing right now, this is going to stain. We're looking to identify the difference between collagen and smooth muscle. So right now what it's doing is it's just preparing the cells to go into their next solution, which is a form of hemotoxin. And then it's going to go into another solution that's a combination solution. This is the quick method. The regular method takes about 6 hours to do. We're going to do it in about 12 minutes. Excluding the hour that you guys haven't seen. So it's going to go into a solution that contains beaver scarlet red, phosphotungstic acid, and aniline blue. And that's going to stain the muscle. The beaver scarlet's going to stain the muscle, and the aniline blue is going to stain the collagen fibers. So we're going to go over there because it's a little more roomy. And we'll start with the fungus. You guys want to grab some gloves? The trichrome stain. So if you turn both of those water spigots on, not super high. One thing that we want to make sure is that the water isn't going to really hit the slides hard, because that tissue is on there, it's adhering to it, but we don't want to kind of be really mean to it. Be gentle with things. Okay, so I have two different types of tissue. I have uterus, and I have small bowel. So the trichrome stain will pretty much work with any tissue in the entire body because we've got smooth muscle everywhere. The types of tissue that we're going to really be looking for things, liver, we're looking for cirrhosis, and colon, we're looking for collagenous colicus. Those are the two big uses for this stain. So going to
We got so excited.
So I'm going to give you each a slide. And if you guys just want to rinse your
Francesco, as the summer goes,
Slides
what I
off.
would do before your contract's all right for the summer, I will go and harvest a bunch of
So these are small bowel if anybody has a preference.
trees and that will see
Oh, it sounds like vinegar.
Yeah, so it's the acetic acid.
Oh.
Yep, you can rinse off the yellow.
If not, yeah, I just put so I had to cut it on Thursday and Friday. We have the first slide, then 2 seconds later.
Does everybody have one? Yes. Okay. That's okay.
Not bad because at least it's not like you have to get into the fridge right away.
I don't know.
You breaking our stuff?
Sorry.
I'm cutting everything.
So for these, since these are going to sit for about 7 minutes,
Okay.
I'm multitasking.
Nice.
You can leave it running. We're going to use it. Okay, so you guys want to grab? Okay. So, okay. So this solution here is got hematoxylin in it. It's an iron hematoxylin, and it's got a beaver scarlet in it. So if you guys want to take that and just squirt it over the tissue.
I really took one.
Yeah,
I think theirs is that one. I'll take the second to last one.
I know this one. I think this one's
Okay. I'll take the third to last one then.
Wait, how do I know this is the right side?
Actually, that's a good question.
I really hope
I don't know
this
if they
is
mind.
okay.
How do we know which one is? Oh,
Like the white bar. You can tell which side you can write on.
are all of ours facing?
If you want to write, sometimes it's easier if you want to write, or you put your initials on there or something. Kind of helps, all of ours are labeled with patient information, so you can tell which is which.
Wait,
Could I use a marker or no?
You're like the middle one,
I would use
and
a
mine's the one in the
pencil.
middle.
Wait, which one's the right side?
So yours is fine. I think they might all be fine. This one might.
Okay.
Here's some. That one was fine. Sorry. Yep, I think. Actually, here's. Yeah. Okay. Try that side. Okay. So while those are cooking, we're gonna start the fungus stain. So you're doing the same thing. I've got a couple different kinds.
Oh, you want to write
You can.
And then if you want to lay them down the same way, but over there.
What is this?
So we're going to start a fungus stain.
Oh, that's good. That's worse than other.
Alright. Well, I was wiping it up. So like I said, the first step, we want to oxidize any of those aldehyde groups that are in the cell walls. So we're going to use periodic acid. So you can guys go ahead and do the same thing that you did over there with the periodic acid.
Please do not get that news.
Clear part.
Okay.
Pretty much guaranteed to
Okay.
cover it at that point.
Yeah, I did.
Janae.
Sorry. Attempt this again.
They cannot work in the lab.
Is it okay that it's got some water on it?
Yeah, it's fine.
I'll make fun of just like my video. We just start this all over again. Whenever stop. But like put on off. That's okay. Attempt number 2. It was even exactly.
Alright, she's complete.
My hands were
Did you not work on it?
I did.
Okay. It was very powerful.
Success. I'll just put that as a video of me dropping that in the sink. It's okay.
Alright, so in the histology lab, we fix everything in formaldehyde. There's a lot of other fixatives that can be used that'll do the same thing in different capacities. So one fixative that can be used is glutaraldehyde. If a tissue is fixed in glutaraldehyde, this stain cannot be done because those aldehyde groups will stain everything that's left from glutaraldehyde. So it's going to stain everything, fungus or not. So as you like work in the lab and you know what's staining what, you'll sort of learn what can and cannot happen beforehand. So glutaraldehyde and a periodic shift fungus reaction is not compatible.